right, we're recording. I guess the first announcement, like I said earlier, because this one's relevant to everybody online, but nobody who's going to be here in person. You still have to email me for attendance today or any other day that I say so. I've had some issues where people, they're here and then I recognize that they're here, like I say good morning or say something about the points. And then I guess maybe they think because of that they don't have to send me the email for attendance. And then because I'm just looking at my attendance sheet and there's people who they were here when the time came, but they never sent me the email. So again, make sure you send me the emails with the keywords. Yeah, I guess that's it. The other announcement for those of you online, it's relevant for you guys and nobody else, is that apparently my audio is not working. I have confirmed that you guys can hear me, but I cannot hear you. So if you have to say anything to me, there's a there's a button on the bottom right that is how you type. You type to chat, and that's how you'll have to communicate with me, I guess. Of course, this has happened before, and then all of a sudden, magically, it started working. So we shall see. So, does anybody want to type some questions before we get started? It is 8 o'clock. All right. As of right now, it's only an online class, so this should be pretty easy. Excuse me. Some other announcements. Independent work. I know I keep saying this, but plagiarism is a big deal. I think the one thing that people know about plagiarism, the easy thing to avoid for plagiarism is don't copy, right? You just don't copy and paste it and say this is mine. Another type of plagiarism, at least in my book, is if you reword something. And I keep saying this, but I think maybe I found a better way to describe what I'm trying to say here. So if you are writing a paper on an article that you've read, forget the words. If it is sentence for sentence the same paper, then that's plagiarism. Because this is what's happening, is people are just changing the words around, and it's still plagiarism. It might be like the first sentence might say something like, this paper is about opioids, right? And they just reword it, but that's still the same first sentence. And then the second sentence is, doctors at this university did this study in 2012. And then again, then when they wrote it, the second sentence basically says the same thing, just in different words. So that's a good way to check yourself. If your paper that you're writing says the same thing, sentence for sentence, that your source says, I just won't count it. It's plagiarism. So it's not just about changing the words. So it might be word for word different, but if it's sentence for sentence the same thing, it's wrong. And for each independent worksheet that I give you, I have a little link you can click where I give you an example of an article and a right way to summarize it and a wrong way to summarize it. So check it out. Again, hopefully this is the last time I have to say this. But the point here is for you to be able to read something, however many times you need to, comprehend it, memorize the important points, and then write about it, right? I'm not asking you to give me, this is not a research paper necessarily, right, where you have to give me all the details you can. I just want you to be able to comprehend something and explain it to somebody. It's a very important skill, especially these days, in the days of misinformation, when all this crap is being shared on social media. You need the skill of being able to read something and actually comprehend what it says. And then again, like I keep saying, and then explain it to somebody else properly. So yeah, that's that. If you guys have any questions about it, let me know. The other announcements are lab. If you had lab on Monday, but you weren't there for some reason, you should come on Wednesday, because to remind you, if you guys do not come to lab, but you still do a lab, like you get the data from your classmates or something, it's only quarter credit, like 1/4 of the credit. So you need to come. For that matter, for those of you guys who were there on Monday, if you feel like coming in on today, obviously you already have your data, but if you want to get another sit down and another hands-on experience with the experiments, then feel free to come over and do that. And then I guess the last reminder for the lab, as you're working on your lab report and doing the lab, you can watch that pre-lab lecture. Like if you still have questions, well, obviously you can come to me, but if you don't feel like coming to me, you can also go to the pre-lab lecture video. Woo! Any questions? Took longer than I wanted. All right. So then let me share my screen. And Schmidt, when you get a chance, do you mind emailing me and tell me that you were here on time? Thanks. Let's see. I would ask somebody to confirm that you can see and hear me, but again, my audio is not working, so I'm just going to have to roll with it. All right. Here's where we left off. We were talking about the basics. Before we even got into the stages of photosynthesis, we were talking about some background information you needed to know. And the last thing we were talking about was the fact how pigments, when they get hit by photons, that excites electrons, right? It energizes them, and they get to this excited state. And they can't stay that way long, and eventually they have to drop back down to the regular state. And when they drop back down, remember, energy can't be created or destroyed, so that energy is released. Very often that energy is released as heat. Sometimes it's used or released in a way that we're going to talk about for photosynthesis. Sometimes it's even released as fluorescence. Like, I'm sure you guys have seen things that glow in the dark, right? And to get them to work, you hold them up to a light, right? And that's what's happening, is when you're holding it up to a light, you're exciting those electrons. You're charging it up, so to speak. But again, sometimes, like I said, sometimes it puts off light. For the purposes of this exam, actually for this entire semester, this is useless information. I'm going to put an X through it. Your book included it, and so did I. And that's the only reason that I included it. So yeah, those little glow sticks, that's how they work. Is I'm sure you've used them before. They're not glowing, and then you break them, and then they glow. Well, what happens is when you break them, it mixes two chemicals, and those two chemicals excite each other, the electrons specifically. And as those electrons lose their excited state, they put off a form of energy known as light. But again, for the purposes of this whole entire semester, that is a useless bit of information. In the thylakoid membrane, which, to remind you, is the part inside of the chloroplast, we have chlorophyll molecules, and they are organized with other molecules into something called photosystems. Now, I'm probably not going to ask you what a photosystem is necessarily, but I will be using this word, so you need to understand what it is. And there's also two different photosystems. And in the exam, you might need to distinguish between the two photosystems. Sort of like in the exam, you need to distinguish between the difference between, mitochondria and a chloroplast, because the mitochondria is where respiration happens, cellular respiration, and the chloroplast is where photosynthesis happens. So the same way, you might need to distinguish between the two photosystems. And I'll come back to that in a second. Your book does point out that each photosystem does have a few hundred pigment molecules. I'm going to put an X through that too. Not because it's not important, but I would just say it's not important for you. You could skip that bullet point and completely understand everything I'm about to teach you. So obviously that should tell you that it's also not going to be on the exam. It was just in your book, so I included it. Any questions so far? And again, we're not done talking about photosystems. So if you're wondering, wait, what is a photosystem? That part's coming. Here's a picture of what I'm talking about. Again, that big thing right here is the chloroplast. We're zooming in because inside that chloroplast are these thylakoid membranes, and you can see they have a phospholipid bilayer, sort of like our cell. And then you can see this little green thing right here. This is a photosystem. I'm going to abbreviate it PS. That is a photosystem. And it's got a bunch of molecules, but we're going to only talk about the ones that are relevant to what you need to know to understand how photosynthesis works. Specifically, one of the most important ones for your understanding would be these little green dots, which are the pigment molecules. Okay. This cluster, these photosystems, they're basically light gathering antennas. So if if photosynthesis is a sugar factory, which essentially it is, it makes, well, we keep saying glucose, but you'll learn later it's not exactly that simple. But anyway, if photosynthesis is a factory that produces glucose or organic molecules, we'll say that, then the photosystems are, let's say they are the solar panels, if you will, that provide the energy for this to happen. What happens is the photon from the sun, it strikes the pigment. From that, the energy jumps from molecule to molecule, because again, when a photon hits a pigment, like we just got finished saying, it excites the electrons in that pigment. And that excitement, in this case, instead of just staying in that one molecule, it just bounces around until finally it arrives at the reaction center. As far as I'm concerned, that part's the most important. I don't even care that you know that it hits one molecule and then bounces around until finally it hits the reaction center. The important thing here is that eventually or even in your mind if you want to think of it immediately, I don't care. Eventually, an excited electron arrives at the reaction center. What is the reaction center? It consists of chlorophyll A, and as you learned, I think Monday, chlorophyll A is the main pigment in this process. And I told you didn't need to memorize that, but there you go. That's back again, chlorophyll A being the main pigment. That excited electron hits the chlorophyll A, and that thing sits right next to something called the primary electron acceptor. That should be in bold as well, primary electron acceptor. And that thing actually traps the excited electron. So again, to go back to what we talked about Monday, a long time ago, I gave you an overview of photosynthesis. And we talked about the two stages in general. Talked about the light reactions and the dark reactions, aka the Calvin cycle. Now, what I told you about the light reactions was, remember, light is a form of energy. So what you should remember about the light reaction, it's all about harvesting the sun's energy and providing a type of energy that the cells can use later in the dark reactions to actually make the sugar. So that would make sense then that we would have this reaction center with a primary electron acceptor that is capturing these excited electrons. Because again, in the light reactions, what we're doing is we are exciting electrons using photons, and we're using those electrons, right? Or we're capturing them because we're going to use them later. So it would make sense that we need something to accept that excited electron. And then there's another team of molecules that traps the energy to make ATP and then ATPH. And remember, like I said, the light reactions are the ones that are harvesting the sun energy and converting it to another form. And then the Calvin cycle is the thing that uses the products, which is the ATP and the NADPH, those high energy electrons, to make glucose. So I know this slide has a lot of information, but essentially this is just an introduction, because all this information is about to come back to you as we move forward, because we still haven't even gotten into the details of light reactions and the Calvin cycle yet. This is still the introduction. This is still giving you background information of what you're going to need to understand those two reactions. So this information, I know it seems like a lot, but we're coming back to it. Otherwise, I would say, do you have any questions about this? But I'm not going to ask you that yet, because again, after we cover it again, that's when I'll ask you if you have questions. The first attendance word for the day, speaking of which, will be question. Like the thing I'm not asking if you have any right now. Question. All right, I'm going to give it a couple more seconds. I see note taking. I don't want to rush anybody. But again, we're coming back to all this information. Here's basically a picture of what we're saying. We already saw this left-hand picture. There's the chloroplast, there's the thylakoids. We zoom into these thylakoids, we can see this photosystem with the molecules, the pigment molecules in there. Again, a photon hits them. That excites the electrons, and the excitement just bounces around randomly until finally it hits the center. It hits the reaction center. And again, as far as I'm concerned, this extra bouncing around stuff is just extra information. That's not important to you guys. What's important here is that a photon is hitting this photosystem. It's exciting an electron at the reaction center, and that electron is then captured by something called the primary electron acceptor. Now, here's the good news. I'm about to put some X's in here. Like I already said, wait, where are we at? I already told you I don't really care about the fact that it bounces around, so I'm going to put an X to that to remind you when you're studying, don't focus in on that. I'm going to put a question mark next to reaction center, because I want you to know that that excited electron has to go somewhere, that I don't necessarily know if I care about that detail of you knowing the name of it. But again, photons are hitting electrons. Electrons are getting excited. They have to go somewhere. Speaking of which, there's something that accepts it. And I'm going to put a question mark through that too, the primary electron acceptor. You need to know that that excited electron goes somewhere, but I'm not sure yet if I care that you know the name of it. I certainly don't care that you know that it's a chlorophyll A that's at the reaction center. If you do know it, great. The more you know, the better. But I don't care that you know that it's chlorophyll A versus chlorophyll B. versus chlorophyll C, which is something we've not talked about because it's not really relevant to most of the things we talk about, or carotenoids or whatever else. So now, are there any questions about this? And we're still going to come back to all this. So this is still just an introduction, but are there any questions so far? All right, let's move forward. Here we go. Yeah, now we're into the good part. Now we're going to learn exactly how the light reactions work. And like I've already told you, most of this exam for Chapter 7 is going to be all about in which stage does this happen? In which stage does that happen? In which stage is this produced? In which stage is that produced? And again, to remind you from Monday, respiration, well, from prior to Monday, respiration has 3 stages. Photosynthesis has 2 stages. So as you're studying this and as I'm going through this, understand that that's most of what the questions are going to be. Which stage produces glucose? Which stage uses carbon dioxide? Which stage makes ATP? Which stage uses ATP? All that stuff. So keep that in the back of your mind as we move forward, as we finally get into the light reactions. So previously, before we talked about that, I introduced you to the concept of photosystems. Turns out there's actually 2 photosystems. And here we go. I hate to be so ambiguous, but I'm going to put another question mark right there. I'm not quite sure that I want you to know that level of detail. Important thing you know here is that we're using photons to excite electrons. Those excited electrons are going to be used to make ATP, and I'm going to tell you how here soon. And those excited electrons are going to be captured and incorporated into the glucose. And I'm going to tell you about that later too. And the fact that there's 2 photosystems, that's almost definitely not going to be a question. Like specifically, how many photosystems are there? That's almost definitely not going to be a question. What I'm not sure about as far as questions are concerned is if I'm going to have you memorize which comes first, and you'll see what I mean. Because again, we're exciting electrons. Some of them, sometimes we'll use it to make ATP. Sometimes we'll use it to make NADPH, and I'm not quite sure if I care that you memorize which order it happens in. Anyway, what happens in these 2 photosystems? The photons excite the electrons, like we already said, in the chlorophyll of the first photosystem. That's the one, that's that picture we already saw. Photon hits the electrons, that excitement bounces around until finally it hits the chlorophyll A. And at that point, that's in the reaction center, and the electron is trapped by the primary electron acceptor. And again, like I said on the picture, I'm not quite sure if I care that you know the name of that. Just know that the photons get excited and they get captured. Here's an important part that we haven't said yet today. Like I said here, we've excited electrons until they bounce around, whatever. And then we've actually trapped it. We've taken it out of the system. So if you take an electron away, then you have to replace it. So here's an important thing you do need to know is where those electrons come from. So as you pull those electrons away, then water gives us the new one. Water replaces it. As you take an electron away, water says, wait, here's another one. So that will be a question. Just like I said in respiration, you have to tell me where the electrons come from and where they go. In respiration, they ultimately came from glucose and ultimately went to oxygen. In photosynthesis, these electrons ultimately came from water. So that's a test question. Another test question is, in which of the 2 stages of photosynthesis is water used? And the answer is the light reactions. Now then, once that water gives up those electrons, that then produces something called oxygen. And like I said, when we talked about respiration, these things go hand in hand, whether we're talking about photosynthesis or respiration. You should think electrons, oxygen, and water. Because in respiration, excuse me, in respiration, the oxygen took those electrons and then became water. Here, the water gives those electrons and becomes oxygen. So it's really just the opposite process of what happens in the respiration. But still, those 3 things are linked together: electrons, water, and oxygen. And that will be a test question too. The first one, again, where do the electrons come from in photosynthesis? Water. And then the second question, follow-up question to that will be, when that molecule gives up the electrons, then what is produced? And the answer is oxygen. Excuse me. Sorry. Another test question that's on this slide. Which of the two stages of photosynthesis produces oxygen? And the answer is the light reactions. And both of those questions will be on there. And when you think about it, one of them gives the other one away. If I'm asking you which molecule is releasing electrons and then producing oxygen, you should think, okay, if we're giving away electrons, that's the light reactions. That's that energetic part. So if you can remember that, then you can remember that oxygen is produced in the light reactions. Any questions about this slide? If I remember correctly, the next slide I'm going to show you is just pictures of all the words that are on this slide. It is indeed. So again, I like this picture better than the other ones I've shown you because it doesn't show all the bouncing around, because in my opinion, that's not important. The light hits the reaction center chlorophyll. It excites the electron. And then that electron is then taken away by the primary electron acceptor. So again, if an electron leaves, you have to replace it with something. What is it replaced with? It's replaced with water. Water says, here, take an electron. And when that happens, that produces oxygen. And you'll notice, just like when we were talking about respiration, there's a bunch of numbers. And I'm not concerned with these numbers. I don't care that you know that it was 2 electrons, right? Just know that there was electrons being excited, electrons being replaced. I don't care that you know that when this water gives up those electrons, and it's half a molecule of oxygen and 2 protons. I don't care about those numbers. I don't even care about the proton part. Again, in photosynthesis, water is the thing that's donating the electrons. And when it does, that creates oxygen. Any questions so far? All right, like I said on the previous slide, there are 2 photosystems being used here. And this is the first photosystem. First photosystem is the part where we're not even quite done talking about the first photosystem, but this is the beginning of it. We've excited these electrons. They've been harvested, so to speak. Let's move forward. In the second photosystem, that energized electron that we were talking about that was just captured in the first one, it then goes down an electron transport chain on its way to the second photosystem. The good news is, if you've already studied respiration, as far as you're concerned, the electron transport chain for this and the electron transport chain for respiration are essentially the same thing. They work basically doing the same thing. Here's a good question, and hopefully you guys remember it, even though I realize the exam's not upcoming, so if you haven't studied it, I get it. What does the electron transport chain, well, let me think this through. It produces a lot. What's the main thing that the electron transport chain produces in respiration? Because yes, it produces water. Yes, it uses oxygen. Yes, it pumps hydrogen ions against the concentration gradient. But what's the big thing the electron transport chain makes in respiration? That's a great test question. And if you're online, I'm sorry, I can't hear you. So do any of you in-person people know? All right, let me reword it to an answer. I swear I will cry myself to sleep tonight if no one gets this. Maybe I was a little bit too specific. The question was, what's the main thing that the electron transport chain produces in respiration? So let me ask you this.
ATP.
Oh, there it is. ATP, yes. I was going to reword it to an even simpler question and say, what's the main thing that we're producing in respiration? Yes, ATP. Yes, the main purpose of the electron transport chain is to produce ATP. So that's for respiration. And guess what? It's the same story here in photosynthesis. We're just trying to produce ATP. Anyway, like I said, it happens the same way it happens in respiration. How is that? Well, that's this bullet point right here. Because they're high energized, right? They're energized electrons from getting hit by a photon. They have high energy. But just like in respiration, it doesn't hold that energy. It falls. As it goes from step to step, it falls, meaning it releases that energy. And that released energy is used to make the ATP. The same way it is, and I'm going to show you a picture of it soon, the same way it is in respiration. That loss of energy is used to pump hydrogen ions against their concentration gradient. So then they're really concentrated on one side and not the other. And then diffusion just happens, and they go from that area of high concentration to low concentration. And as they do, they go through the enzyme, that produces ATP. I'm saying that pretty quick right now because you're already familiar with it from respiration and because I'm about to show you a picture of it. So anyway, yes, we get the excited electron. It goes through the electron transport chain. That produces ATP. And then the second photosystem transfers that light excited electron to the NADP+, converting it to NADPH. And I'm going to put an X through this. Of course, this is true. It does convert it to NADPH. But like I said, when I was talking about photosynthesis or respiration and photosynthesis, when you see a molecule that looks like that, whether it's NADPH or FADH2, whatever, if it looks like that, you should just think that's the thing that's transferring the electrons from one place to the other. So again, all this information that's on the board right now, I'm about to show you a picture of, and hopefully it'll make a lot more sense. The next word for attendance is going to be vacation. I'm already thinking about spring break. Not that it's going to be vacation. I'm sure my wife's going to put me to work, and I'll be grading papers and stuff, but still. Here we go. Here's a picture of everything we said. Photosystem 1, photosystem 2. Again, photons hit photosystem 1. It excites the electrons. The electrons leave, and of course, they've got to be replaced, which is where the water comes in. Water says, here's some electrons. That produces oxygen. That excited electron then goes from protein to protein down the electron transport chain. That energy is used to make ATP. And again, you're familiar with this because it's pretty much the same thing that happens in respiration. And I'm about to show you yet another picture of this. So I'm going to come back to this concept again. Anyway, so as it's going down that electron transport chain and releasing its energy to pump hydrogen ions against the concentration gradient, it's again at a lower state, right? It's not energized anymore. It lost its energy. So then it goes to the second photosystem where light hits it again and it gets energized again. And it gets captured again by a primary electron acceptor, except this time, instead of going down another electron transport chain, it's captured by those things that just carry around the electrons from one place to the next. So at this point, we have exactly what we need to make glucose in the Calvin cycle. Well, a portion. We have the energy that we need, right? We have, where are we at? There we go. We have the ATP, the energy that we need to squish those carbon molecules together. And also now we have some high energy electrons to incorporate into those molecules of glucose. Excuse me. So are there any questions about what's on this slide? All right. So earlier I said I put a question mark between photosystem 1 and photosystem 2. I said I wasn't sure if I was going to ask you to know the difference, like the sequence of which came first, photosystem 1 or photosystem 2. And I'm probably not going to specifically ask you. But when you're looking at it, when you think about it, it can only be in this way. Because if in photosystem 1, we captured the electrons and brought them off on with the NADPH, then that would be the end of it, right? Then we wouldn't have, that thing would be gone and take those electrons somewhere else. So without photosystem 1 giving us the electron transport chain, right? If it went directly to this, then we wouldn't even have the electron transport chain. So yes, I'm probably not going to ask you the order, but logically it can only happen in that sequence. You cannot have the NADPH come first because then the electrons would just be gone. They would have never gone through the electron transport chain. You would have never had the ATP. Any questions? All right. I like this picture from your book. It shows the same concepts, but then it changes it up a little bit. So again, you've got this electron that's not excited, and then you have a photon that's exciting it. But in this case, the photon is represented by somebody with a big old sledgehammer. They hit that lever, that thing bounces up. You got the primary electron acceptor that takes it, that excited electron. High energy electron. In this case, you might high energy and excited are essentially the same thing, but in this context, I like the word high energy better because now it has all this potential energy, right? Like in real life, a ball up high would have potential energy. And what would happen if you were to drop that ball? It would roll downhill. And that rolling down the hill could turn a turbine, which in this case would produce ATP. And then again, it gets down here where it doesn't have as much potential energy, right? Because it's gone down. So photon uses some kinetic energy, boom, hits it again. And again, it's captured again by a different electron acceptor. And now it's in this bucket. It's ready to be moved somewhere else. And it's still up high, right? So it still has these electrons, or this electron here, has more potential energy than these electrons, and definitely has more potential than that first electron, right? So again, think about what we talked about in chapter 5, kinetic and potential energy. Now, granted, in real life, when we're talking about electrons, it's not necessarily kinetic and potential energy, but it's the same concept. It's about excited and not excited. All right, any questions about this picture? All right, another thing I like about this picture too is it doesn't have the numbers, because remember, I don't care that you know that it's technically 2 electrons that go. But anyway, excuse me. Okay, like I've already said, the ATP production is similar to the way it is in respiration, because in both of them, the electron transport chain is pumping hydrogen ions, AKA, I usually don't do this, but I've been watching that video that I shared with you guys from Hank. He uses the word proton, and that's the same thing. Hydrogen ion is a proton, so I'll use both words to make sure you understand what I'm talking about. In the exam, though, you should expect to see the word hydrogen ions or the H+, because that's what's in your textbook. Anyway, like I said, the electron transport chain is pumping hydrogen ions against their concentration gradient. Of course, it takes energy to do that because it's going against the concentration gradient. You're going from an area of low to high concentration. And like we said in chapter 5, that requires energy. That energy is coming from the electrons in the electron transport chain. And then, of course, those protons, those hydrogen ions, they want to go back down their concentration gradient. So they do. They diffuse down. And then they do it through the same enzyme, too. Notice that. ATP synthase is the enzyme that we're using in both electron transport chains. Whether we're talking about photosynthesis or respiration, it is ATP synthase is the enzyme that's doing it. But here's the main difference. I keep saying electrons have to come from somewhere, they have to go from somewhere. If we're talking about cellular respiration, those high energy electrons, they came from food. Specifically, glucose is the one we focus on, right? Glucose is the thing that provided those high energy electrons. Because even in the first step, glycolysis, we broke glucose down into pyruvate. We stripped away high energy electrons. And then we broke that down into acetic acid and stripped away high energy electrons. And then we put that thing in the Calvin cycle and broke it down even more and stripped away high energy electrons. So in respiration, those high energy electrons came from glucose or food. Food is a better word when it comes to just thinking about how it works, because obviously you guys eat a lot more than just glucose. But as far as the exam is concerned, the word glucose will be better, because that's what we're focusing on, because I want to keep it simple. Well, as simple as I can. Glucose provides high energy electrons. So again, that's respiration. And photosynthesis, it's the water that provides those electrons. The water is like, here, take some electrons. So any questions about that side? All right. Here's a picture of it all in action. Lights hit that photosystem. Electrons are excited. And then as they go from protein to protein in the electron transport chain, they're giving off that energy that they have. That energy is used to pump hydrogen ions against their concentration gradient. Eventually, they're just going to diffuse back down their concentration gradient. And as they do, they're going to do that through this enzyme called ATP synthase. And that is the thing that's going to be producing the ATP. Meanwhile, after that electron went from protein to protein and hit photosystem 2, again, it gets excited by the photons, and then that gets captured. So the big picture stuff, the most important thing here is that we have ATP and these high energy electrons carried by the NADPH have been created, because those are the two things that are going in the Calvin cycle. So as far as the big picture is concerned, that's the most important thing. Because the Calvin cycle is the thing that's actually making the glucose. And how does it work? It's using the energy from this from the light reactions, specifically the high energy electrons carried in the NADPH and the ATP. Any questions about this side? All right. And here's a picture that you should already be familiar with. That's the one from respiration. And then look, it's the same thing essentially, right? You have electrons going from protein to protein to protein in the electron transport chain. That's pumping hydrogen ions, aka protons, against their concentration gradient. Eventually, they just diffuse back down their concentration gradient through this enzyme called ATP synthase, and that produces ATP. So similar thing here, right? I mean, as far as you're concerned, at this level, it is the same thing. Electrons pumping high energy electrons pumping protons against their concentration gradient so they can diffuse back down and produce ATP. So that's it for the light reactions. And next we'll talk about the Calvin cycle, aka the dark reactions. And again, remember, for this exam, most of the questions are going to be, in which stage does this happen? In which stage does that happen? In which stage is this used? In which stage is that produced? So basically, if you guys were really on top of your game right now, I could give you questions about the Calvin cycle, and you should be able to answer them because you would know how the process of elimination. Like if I said, which stage uses ATP? Hopefully you would know it's the Calvin cycle because we just got done talking about how the light reactions produce ATP. Likewise, if I said, which stage uses high energy electrons from NADPH? That's the Calvin cycle, right? Because we just got finished talking about how it's the light reactions that produce it. Or if I said, which of the two stages uses carbon dioxide? Hopefully you could use your brains and think, well, we've already talked about one of them and we never mentioned anything about carbon dioxide. So therefore, the Calvin cycle must be the answer to that question. So anyway, keep all that in mind as we go forward. That's mostly how the test questions are going to be. Calvin cycle. This is how I like to think of it. It's a sugar factory. This is where we finally make our glucose. Just like any other factory, it requires ingredients. Well, just like any factory that's making something edible, it requires ingredients and it requires energy to put those things together. This bullet point on the second, not very important, or not important at all as far as the exam is concerned. Yes, it has a starting material that I'm going to explain. That starting material is regenerated every time, hence the name cycle. But again, for the exam, that's not important. If you watch that video I showed you with Hank, he talks about it in more detail than I will. Each chemical turn, each turn of the chemical process, the cycle, it has inputs, it has outputs. We'll talk about them, but again, I'm putting an X to it because I'm trying to keep this as simple as I can. So for the exam, we won't talk about that unless otherwise noted. Yes, I get a sense you do need to know the inputs and outputs. But the question is not going to be, what are the inputs of the Calvin cycle? What are the products of the Calvin cycle? But I will say, and there's some test questions on this slide that I'm going to come back to in a second. Which of the two stages of photosynthesis uses carbon dioxide? And the answer is the Calvin cycle. Which of the two stages of photosynthesis uses ATP? Not produces, uses. Someone's going to miss that. And the answer is the Calvin cycle. Which of those two stages uses high energy electrons being carried by NADPH? Calvin cycle. Which of the two stages produces sugar molecules? And I keep saying glucose. We'll come back to that in a second. The answer is Calvin cycle. So again, if you can remember, it's the Calvin cycle that's putting the ingredients together to build what we want. If you can remember that, then obviously, I don't know how obvious it is, but if you can remember that this is the thing that's building what we actually want, then hopefully you can remember that the other process, the light reactions, that's all about the energy. That's the thing that's giving this the energy to do what it has to do. So any questions about what's on this slide? And what we're about to talk about it some more with some illustrations. All right, here's how it works. At least this is the version that your book gives you. It's actually a lot more complicated than what I'm about to show you, but that's okay. So we have this starting material, this RUBP sugar. Notice it has 1, 2, 3, 4, 5 carbons, 2 phosphates. I'm going to say notice because I'm going to talk about it. It won't be on the exam. So anyway, we've got that sugar. What happens is at the beginning of the Calvin cycle, we have this carbon dioxide from the air. That 1 carbon is added to that receptor molecule. Then we have a 3-carbon molecule. And hopefully, if you guys are paying attention, somebody right now is a little bit confused. I hope. No one, I've been teaching this for a while, and I've never had somebody confused at this point, but you should be a little bit. But I just told you that this 5-carbon sugar gets one more carbon right here, and then we end up with this 3-carbon molecule. None of you are math majors, but can someone tell me what 5 plus 1 is? 6, right? So that should tell you something's up. Something's wonky. How do we go from 5 plus 1 to 6? And if I remember correctly, your book doesn't even talk about it in great detail. I'm about to explain what happened. So anyway, we end up with this new 3-carbon molecule. We use a little energy right there via ATP. We incorporate some high energy electrons via NADPH, and we end up with this thing called G3P sugar. But here's where this other electron came from, or the other carbon is accounted for. What happens here is this thing is added to that thing, the RUBP sugar is added to the carbon dioxide, and what you actually have is 2 of these 3-carbon molecules. And then they go through the cycle, they go through this, they go get a track, they do some stuff, some magic that we don't talk about that requires energy. That's where the ATP comes from. We incorporate some high energy electrons to those 2 molecules. That's where the NADPH comes from. And then at this point, we actually have 2 molecules of this G3P sugar. And the only reason that's important is because really that's what we're after, is that sugar. One of them we use, the other one goes back into the cycle. And that G3P sugar is used to make glucose and other organic compounds. So as far as I'm concerned, I'm putting a little X through this, you don't even need to memorize that middle step. As far as I'm concerned, photosynthesis makes glucose. Yes, it has to have the G3P to make the glucose. And yes, that G3P is also used to make other organic compounds, but I just want to keep things simple. And as far as we're concerned for this exam, photosynthesis produces glucose and respiration uses glucose. Right? So anyway, that other G3P sugar, it's back in the system. Whoops, wrong way. We use another ATP to do some more magic to it. And now all of a sudden we end up with the starting material again, the RUBP sugar, and the circle of life continues. the cycle continues. Now, again, this is more detail than you need to know for the exam. What do you need to know from the exam? Which of the 2 stages uses carbon dioxide? Calvin cycle. And remember, I did that on purpose. Calvin cycle, carbon dioxide, all those C's go together. Which of the 2 stages uses, not produces, uses ATP? Calvin cycle. Which of the 2 stages uses high energy electrons being carried by NADPH? Again, uses, not produces. Calvin cycle. Which of the 2 stages produces glucose? Calvin cycle. And that's it for the exam on what's on this picture. I'm not going to ask you about G3P sugar. I'm not going to ask you where the ATP is used or where the high energy electrons are used. You just need to know that they're used. Any questions about that picture? And again, all this is happening inside the mitochondria. I like that picture. It shows everything. Again, light reaction to the thing producing the high energy electrons and producing the ATP. And the Calvin cycle is using those things to squishy a bunch of carbons together to make glucose, to make sugar. Pretty simple. Review. If you download the PowerPoint, you can look at this review. It's everything we just said. It's all together. We're going through it again. I prefer just to end 8 minutes early to make up for last Friday when I kept you guys late. Even though you did get extra points for it, I still want to make up for it. So does anybody have any questions? All right. Schmidt was here on time. Make sure you email me to tell me you were here on time. You guys make sure to email me, say you were late. You kind of had both going. So when you email me, use the attendance or the keywords from the point that you were not here. Okay. All right. And that's it. I will see you guys at office hours.
Are we only doing 2 keywords?
What was the question?
Are we only doing 2 keywords?
I think I said, did I only do 2? I would be fine with that, but let's do another one because if you only do 2, then if someone misses one, then they have a horrible grade. Now, let's say the next keyword is pencil sharpener. Well, remind me that on Friday. Yeah, so the next word is pencil sharpener.
All right.
All right.
Thank you.
That's it. Remember to email me, guys. I just looked over there. Yeah. If you want like all the points, when I put out the video, you can just watch the video and do the questions. It's up to you. Hamrick, are you still here?